It's a real pleasure to have with us a very energetic and innovative spin-off startup from Greece, Purposeful, who is employing data integration and artificial intelligence methods for a very challenging problem, which is the task of repurposing drugs to new indications and particularly unmet needs in rare diseases. I think this is going to be a very interesting presentation for many of us. And George, thank you for accepting our invitation. The floor is yours.
George, thank you very much for the introduction. It's a pleasure for Purposeful to be able to speak here as well. I'm going to introduce our company. As George mentioned, we are a startup which we focus on repurposing drugs in rare diseases. So maybe a couple of minor definitions before we get into the good stuff. So the idea of repurposing has to do with using existing agents, commonly drugs, sometimes nutraceuticals, which are already on the marketed, safe for the treatment of other disorders for which are previously unknown treatment. As you can see here at the top, we are primarily focused on rare diseases. So it's not only rare diseases, but that is 90% of our focus, probably. So what is a rare disease? It's a disease that is documented to affect 1 in 2,000 people, or 1 in 3,000 people, or 1 in 5,000 people. So the larger the denominator, the more rare the disease. And first question we always get is, why do we focus on rare? So there's lots of reasons. The lack of treatments is one, for which I will speak to a little bit later. And the second thing is the feasibility for a very small new entity in Greece. So it is an endeavor that is obviously less costly, and due to the fact that it's not affecting so many people, is not of interest to large pharma companies, so there's less competition there as well. So let me talk a little bit about the problem we're talking about. We have 7,000 rare diseases, give or take, of which 95% do not have a known cure or treatment. It affects over 400 million people worldwide. So you might think rare disease, I gave you some numbers before, but when you put them all together, it's not that rare. There's estimates up to 1 in 10 or 1 in 20. The literature differs, but there's estimates that they're not as common when you take them all together. And one other point is that half of the people affected by rare disease are children. And now let's move into some other problematic numbers. When you try and do de novo drug discovery, so this means to develop a new agent, the average cost, according to the literature, is over $2.5 billion. And the discovery timeline is a bit bleak as well, averaging at about 13.5 years. So especially when we come to rare diseases, which is our primary focus, in, I'd say, the majority, if not all the cases, de novo drug discovery isn't really a viable option. So what do we do? How is our solution contributing to this, right? So we've talked about drug repurposing, what it is, but there's a couple of key points I'd like to go through here. First of all, we're talking about drugs that have been extensively tested. So we are aware of their safety profiles, and unless you do something nuts with the dosage or something, they are considered to be safe. The other two numbers I'm going to give you here are literature numbers, so it's not always the case, but drug repurposing does tend to be cost effective. It's estimated to be a fraction of typical drug discovery pipeline, and much faster, so on average about 8 or 8.5 years faster. Of course, these numbers do come from both rare and common diseases, so do with this what you will. Now, let's talk about the other thing that we bring to the table. We are a purely computational team, so we use machine learning, and a few reasons for this. First of all, it's cost effective. So it's quick. We don't need to do as many preclinical experiments. And secondly, we want to bring this robust point to focus, because up until very recently, and even nowadays, a lot of drug discovery and drug repurposing relies on people with domain knowledge, which will look at the molecules and say, this will work, this won't work. And in all fairness, they are right a lot of the time, but this is based on the sort of the patterns that they've formed in their mind, and not on the patterns that the computer can figure out. So we have at least something that does not rely on hunches anymore and could potentially provide novel results as well. Final thing for rare diseases, we discussed that it was cost effective a little bit earlier, but we can sort of qualify it now a little bit. Smaller patient populations means that you have to, by definition, be allowed to do smaller clinical trials. So this brings down the cost a lot. Another thing is that there are several incentives for those who do want to go into rare diseases. There's a process called orphan designation. When you match up a new drug or an existing drug study with an indication with no prior treatment, if you get granted this orphan designation, then there's all sorts of benefits for you. You have an access to a separate grant pool, you get scientific advice, and you're provided with market exclusivity. So at least some of the investment that you've made can sort of, you can have some return. Now, a little bit, I know this is a bit of a loaded slide, but let's try and bring a little bit of simplicity to this. So at the top, you can see sort of a linear process for which we work as a company. So we have from data to filtering or processing, if you will, modeling, then the prioritization of candidates, validation, and finally the utilization. This, as you can see, is somewhat of a linear representation. I want you to think of it also as a stacked representation. So if you have 5 databases, you can make them 15. If you have 100 million models, you can make them 300 million models, and so on. So each of these nodes can benefit from further information. So let's start a little bit with the data. We look at both the drug side and the disease side. So for the drug side, we have a look at bioactivity, gene expression, the biological pathways implicated, and additional information such as side effects. From the disease side, we want to see if there is a genetic component. We have a look at somatic mutations. We look at any reproduced animal models with knockouts. We have a look at drugs, whether they're used properly or off-label or for a very, very small subset of particular phenotypes. We look at RNA-seq data. We also mine the literature, so we do have text mining associations as well. All of this is sort of trying to describe both the small molecules and the disease as well as possible. And then we go into the filtering node. The filtering node we have on the drug side, well, actually, let's break it up with the bullets on the top. So we have a scientific rationale and a business rationale, if you will. So the scientific rationale has to do with what is active. Is 1 micromolar on a GPCR or a kinase the same thing, or do we have to talk about something different? We have a lot of pre-processing trying to be able to cover as much of the chemical space as possible. Same with the biological space. So we do have a sort of in-house custom-built algorithm for doing that. We have to consider other things. If, for example, a drug needs to get into the brain, that we need to have a model or for penetrating the blood-brain barrier. Similarly, on the disease side, we can have a look at similarities between classes, between ontologies. So this is more or less sort of filters / pre-processing for the scientific part. For the business part, we have to, of course, look at whether or not this actually can be utilized. It does have a space in the market. So this, we could look at the prevalence of a particular rare disease, because there are rare diseases which affect a few million people. There's rare diseases which affect 2 or 3 people in the world, and it's a shame. And we'd be happy to make predictions and talk to the physician, but at the same time, there's no way that this can become a business. Same happens when you factor in costs of clinical trials and each disease is very, very different. So we have to make sure that we do have the business rationale before we can proceed to the next node. Next node has to do with our modeling infrastructure. This has two components. We make predictions for the drugs on the market. We predict new properties using several million models. I want to make a side note here and say that we use explainable AI. So we want to be able, when we have a prediction, to actually go back and see why this prediction occurred. So that's something that isn't very common nowadays. And then when we have these new properties, we have to make sure that they are significant enough for a particular disorder. So we perform enrichment calculations for all the disorders in our database. So there's a matchup between roughly 2,500 agents, I think, for which we've predicted new properties, and approximately 13,000 diseases. And then with all of this modeling and this matchup, we try and select the top candidates, which would be possible to repurpose. The next stage is probably the most important, the prioritization. So, disease dependent, you can have 5 predictions, which be fantastic. Sometimes you might have 50 or 100, and then you have to sort of prioritize because you don't have the resources to test all the agents. And this has a couple of components. One is the automatic one, where we have to look to see if something is already said in the literature, because then there's no novelty from our side. Something is already in trials, and so on. So there's an automated component here. And then there's also a manual component, which has to do a little bit with our in-house expertise, with the per disease uniqueness. So do you want a really promiscuous drug? Do you want a really selective drug? Is there a current mechanism that is really trendy? Is there something else that you want to do? Diversity analysis. If you have 5 agents which are antipsychotics and 5 agents which are anti-neoplastics, you don't want to test all 10 of these. You'll choose 1 from each category, and so on. Second component to this is the trying to reach out to world experts. So this has to do a little bit with our network and a little bit with whether or not we have a patient organization on board with us, because they do have experts which they can link us up to. And this is really important. So this sort of feedback that we get can either make us go back and try stuff again, or at least help us prioritize a little bit which agents would make sense for a particular disorder. So once we've gone through this, which, as I said, is probably the most significant step, we have to go and try and validate our findings, because these, at this point, are merely predictions. We find the trusted labs of ideally the patient organization, but otherwise collaborators of ours, and we design the best possible preclinical validation to this. Once we have confirmation, we protect our information and we try and utilize. So what does this mean? This means that we try and prove that this works also in humans, usually with a pilot phase 2A trial. So I think I already mentioned this. Yeah. So what we try to do is basically de-risk the entire process. So after a phase 2 trial, the process, when it's confirmed in people with the particular disorder, the risk of going through a dose finding 2B or a phase 3 trial is minimized, and therefore the market authorization is also minimized. I want to take you through this little graph that I have at the bottom, just to give you a couple of numbers. So the way, I don't know if you can see my cursor, by the way, hopefully you can. So we go from the computational prediction to the post-discovery or the filtering, as we said. Then we have the preclinical validation, either in vitro, so in cells, or the like, in vivo, in an animal model. And then we have a regulatory component to this. So this is sort of a little bit of how do we protect our information, how do we apply for the orphan designation, and how do we design the clinical protocol. So this commonly does come with an investigation of new drug submission, an IND submission. And if this goes through, the authorities, FDA, EMA, or what have you, plus the ethics board of a hospital, then you could be granted the chance to get to a phase 2 trial. And then you have the clinical aspect. So in rare diseases, this is a little bit gray. So sometimes you need a phase 1 trial to establish safety if you're doing a bizarre combination, or if you're doing something outside of known doses, or if you're doing something but people already on several heavy medications. But a lot of the time you can skip the phase 1 trial, which is why it's not in this graph. And then you have phase 2, which can be A and B. This is commonly done if you want a pilot trial and then you want to maybe optimize the dose, or if you have a combination, and for the phase 2A you'd have a fixed dose combination, for the phase 2B you'd have a variable doses, and then the phase 3 is sort of the larger scale one, and then you have another last sort of node here, which would be the market authorization. This is sort of the end-to-end process of our repurposing pipeline. To give you a little bit of information for what this looks like for de novo drug discovery, there's a lot of nodes missing here. So you'd have to screen approximately 10,000 molecules, which you've synthesized, to get some hits. Then you'd have to go from hits to lead. Then the lead, you'd start to modify the chemistry to lead optimization. You'd end up with approximately 500 drugs in the animal model and maybe 5 in the pilot phase 2 trial. So until one will finally make it to the market. This is something which is very, very, very expensive. And in our case, just to give you, because I told you it was 10,000 drugs, in our case, I think the most we've tested so far has been 10 agents. So you can kind of get the idea of how much less resources are used for this. So in our pipeline, we have several disorders. I can't list them all. I just want to show you that there's different sort of maturity levels for these. You might note here that there is one particular disorder which isn't rare. We're aware of this. This is a collaboration with a pharmaceutical partner. So this is something that is also going quite well. But let's have a look. So we have some confirmation in animals for glioblastoma and obsessive compulsive disorder. We have some cell-based assays for rare inflammatory skin disorders. And when I say rare, this is for our aspect. The truth is that the cell-based assays would also give you readouts for more common disorders like psoriasis or eczema and stuff like that. And then there's some others in the pipeline which are just past the post-discovery phase. So Duchenne muscular dystrophy and Pitt-Hopkins. And finally, there's some others which are now currently in negotiation with patient groups. So I'm just showing you different levels of maturity. However, the one I skipped is the one that we want to focus on today as a case study. So this has to do with Fragile X syndrome, which, as you can see, is a lot more advanced, moving towards, well, actually currently in a phase 2 trial. So let's see, this would be the highlight of the last quarter of last year and the first quarter of this year. So we've managed to find a compound combination which seems to, well, alleviates all phenotypes in mice, and it seems to be promising both for the behavioral aspect and for the cognitive aspect of this particular disorder. We were very, very lucky to partner with Fraxa, which is the organization for Fragile X, and we have one of the world leader for Fragile X in terms of protocol writing and sort of the physician for this, Dr. Elizabeth Berry-Kravis, and the medical advisor of Fraxa is Dr. Mector Fanglia. So this is the highlight which I would like to take you through today. First thing, we did use them for bouncing ideas off of when we were refining the candidates list, as I mentioned earlier. They put us in contact with the Drug Validation Initiative, which is the trusted lab. So we were able to know that the readouts from our animal model, we wanted to confirm the potency of our candidates was that readouts were going to be meaningful. And then finally, one of their partners, the Rush Medical Hospital, which is now running the trial for us. So these 3 steps, they seem, you know, like they are sort of very easy to establish, but really this is very difficult, and this is credit to Fraxa, because they are a very organized group and they can get things done quickly. And this is sort of one thing that I do want to say before I talk to you about the protocol, is that this particular clinical trial has been designed and started within, I think, 1.5 years from the idea. So this process can become very, very fast if the right people are involved. So I told you a little bit about the fact that we're doing a trial, but you may be interested to know about what Fragile X is. So the abbreviation is FXS. It's a neurodevelopmental disorder. And again, the numbers here, you can get them from the literature. Usually they say 1 in 4,000 individuals. And there is a couple of effects associated with this. So first of all, there's delayed development of speech and language, and you can usually realize that by the age of 2. But the most common, because there's more symptoms, we have low IQ, we have obsessive compulsive behaviors, social anxiety, hyperactivity, sleep problems, and aggressiveness. These don't all coincide at the same severity all the time, so there's different ages for which one is more severe than the other. But overall, these are the more sort of common and severe symptoms. You do have cases of what am I looking for? I can't find the word in English, unfortunately, but seizures, that's the word. You do have seizures and several other things, but these are the more common ones. And the Fragile X disorder is classified as an autism spectrum disorder as well. So how do you validate any findings that could be useful for this disease? So there's an animal model, because we know the genetic component. It's the FMR1 gene, so when you knock it out, you have issues with FMRP. And you assess 6 tests, which will help you to see whether or not you have improvement in behavior and cognition. So you have the open field test. This has to do with assessing hyperactivity. Basically, the knockout mice tend to be a little bit more active. You have grooming, self-grooming, which this is to monitor hygiene maintenance and other processes. Again, we do have a difference between the wild type and the knockout. The 3-partition test, this has to do with sociability. So you do have the mice, and they can choose whether they would like to spend time with the mouse they've been previously acquainted with, or with a completely novel mouse. Similar test, but not with a living organism. We have novel object recognition, which has to do with the natural curiosity of the mice. So wild type mice do like to spend more time with novel objects than with objects they've seen before, and this difference isn't apparent in the knockout mice. Very interesting test is the hyperneophagia test, for which we have mice on a feeding schedule, and then you change them from the environment and you introduce a new feeding source. This has to do with the they usually have latency for when they will actually start to eat, and they will be very defensive and eat only little quantities. So again, there's a difference between the wild type and the knockout. And finally, from the people performing the experiment, they can also score how well both the wild type and the knockout mice can create a nest. Let's see if we can explain this nice and quickly. We have readouts for these tests. The first sort of column here will be the wild type, and the second column will be the knockout, so the diseased mice. You can see in all of these graphs there's a difference, and what we want to do is we want to be able to reverse these distances and try and bring down to the original wild type. So the other 3, so from the middle and to the right, the other 3 readouts that you're looking at have to do with our drug combination. First of all is one agent, second is the other agent, and then finally we have the drug combination. And you can see in a lot of the cases that the single agent does indeed reverse the phenotypes, but not in all the cases. What we have found is that there is an additive / synergistic effect, and the combination of these 2 agents is the only one that can actually reverse all phenotypes. So I will guide your attention to the last column here, which you will see is highly similar to the wild type, which is exactly what we want to see for all of these tests. Now, obviously, we want to see how this translates to humans. So, as I said, we are going into a phase 2A trial. We are introducing 4 different periods. I'm not going to list these because we have to preserve the sort of the single blind for the participants. But you can imagine that at all times, participants will be taking 2 identical pills, and then if there is one for one agent and one placebo, then one of them will be an over-encapsulated placebo, and the other one will be the over-encapsulated drug. So this will be, this is, not will be, I keep saying this as if it's a future thing. This is the way to make sure that the combination we will be able to assess whether or not the combination is indeed better than a single agent in the human experiment as well. So first things first, we do have to assess safety and tolerability for this. So we do look at adverse events, we look at vital signs. There is blood work and urinalysis involved as well during the visits to the clinic. And we have a series of tests for which translate cognitive and the behavioral phenotypes. So I'll bring your attention to, because I realize that the time is sort of running out, so I'll go through them relatively quickly. We have the KITAP, which has to do with alertness and distractibility. We have the CGI-S and CGI-I. So these two are investigator rating, and this has to do with the severity and the improvement. We have the VAS, ABC, and Adams test. These are for the parent or the caregiver. These have to do with questionnaires and scores for which they can assess all sorts of functioning, anxiety, sleeping patterns, hyperactivity, social avoidance, and so on. We have the iBehavior ratings. This is a smartphone app. This is quite new. And we have the clinician standardized interview as well, which is quite helpful. You know, sometimes the caretakers will not be as objective as the clinicians, so this is always very good to have. The NIH toolbox is standard battery for assessing the cognition. And finally, two very, very much established tests in this area. We have the ERP, so event-related potentials. Here, we essentially look at delta power. So this is the electric signals using an EEG, and the eye tracking. So whether or not people can maintain focus. I think I went through these quickly enough. I will very, very quickly introduce the team. So it consists of myself. My background was in computer sciences, but I moved into computational chemistry for my PhD. I'm going to skip one. I'm going to go to Georgia. Her background is in biostatistics and bioinformatics, all sorts of omics analytics. We have Haralambos, who is the person that puts all of this together, all of the computational infrastructure. He's a software architect in charge of anything from databases to all the services, all the APIs, everything. And finally, we have Aristides Dogmatsidis, excellent expert to have on board, expertise in quantitative clinical pharmacology, pharmacometrics, very, very broad background. So this interdisciplinary team was the core. We are actually very happy that we've just hired the molecular biologist as well, Thalia Vlahu, so another expert joining the team and sort of starting to fill in these little pieces of the puzzle that are missing. And what's next for us? So you may have seen when I showed you the pipeline that we had sort of a wide array of diseases we were looking at, and this was great because we wanted to establish that this methodology works. But now we have to sort of bring it back down again. So we don't want to go any broader. We rather want to focus on the things that have worked. So we are moving in various other neurological disorders and ASD, so autism spectrum disorders as well. So we're looking at Pitt-Hopkins, Angelman, Rett, and so on. We're looking at others for which they do have organized patient associations, such as acrodyssystosis. And I mentioned that we're also doing one in our pipeline, one disorder, which is a common disorder. So we do have collaborations with pharma companies, and it's for more than one disorder. I just can't disclose everything currently. And of course, we want to expand our engines. So we want to be able to constantly add more data, more models, more knowledge, different ways of looking at things. And we have now gone completely autonomous. So instead of relying both on different providers and at the same time third-party APIs as well, now we have everything in-house, which is really, really good news. And I think this is my last slide, for which I want to just say, you know, things maybe to feed the open discussion afterwards. Future needs, I guess I'll start there. Obviously, we always like to have collaborations, and you can see it's a very interdisciplinary process. So there's lots of room. I've just listed a couple of bullets here, but obviously there's lots of other areas of expertise that would be useful for us, and for partnerships as well, because there's lots of preclinical validation that needs to be done. So we need the labs. We are interested, of course, for the later stages as well for co-development, and there's also stuff that we can do in terms of grants and calls, because it's not only our area of focus. Our engine will also work outside of this. So maybe we won't be able to do exactly what we do in an academic setting, but if we switch around the data a little bit, go to experimental molecules or go to natural products or something, this is always something that we can do. Now, I think in the talk description, I also wanted to mention the challenges faced, so I'll go through them quite quickly. So one of them has to do with the network, and this is vague, I know, but it has to do a little bit with, do you have enough experts in your network to bounce ideas off of? Do you have enough labs for when you want to get quotes or when you want to perform preclinical work? And are they able to advise you correctly for what endpoints need to be set, for example? Another thing has to do with the maturity of your company, right? So when 3 years ago, when we didn't even have a single line of code and we pitched the idea, it was very difficult for people to actually agree and sort of participate with us. But as we've been proving ourselves along the line, I can see that this is sort of growing nicely, which is a nice effect. Another thing is the interdisciplinary aspect. As I mentioned, there's still lots of things that we do as a core team, but there's needs for people with expertise in the autism spectrum disorder. There's needs for people with clinical expertise and all sorts. Even for the things that we are the experts in, we still need people to help us with the programming, help us with the modeling, help us with all sorts. Costly, yes. This endeavor is something which is very, very, very expensive. Just to give you an idea, when you're trying to validate in a preclinical model, there's a wide variety, but I want to tell you of a recent quote that we had. So to validate 2 drugs in an animal model against a particular disorder, which will remain nameless, we had a quote for 350,000 euros. You might be able to understand that for a startup in Greece, this isn't something that is sort of very easy to navigate. So, and I don't even want to mention the costs of clinical trials depending on a disease. It's very, very expensive. So you do need people that do believe in your vision and do want to sort of co-develop or invest or what have you. And finally, since I presume there are AI people in the audience, I do want to talk about the importance of data. We use publicly available data. Obviously, lots of private databases in this particular field are very, very expensive. So yeah, but obviously there's also the idea of what type of data can you automate, integrate, because you have structured, unstructured, you have data on the small molecule and how to represent it. You have data on the biological activity. You have all the way to clinical, and there's different ways of representing this. Yeah, just a couple of points for the open discussion, I guess. Thank you very much.
Thank you, George. This was also very interesting. I think was at the right pace. It was nice to see the context, putting AI and the rest of the technologies in context and giving the big picture of getting from the beginning to the end. And I'm really glad to hear that you started the clinical trial.
Thank you.
Any questions?
A little bit about the techniques from ML. Yeah. Well, what can I say without giving away too much from the pipeline? Explainable AI, I think I already mentioned that. So we are talking about non-deep learning currently, although we do have a pilot going for deep learning as well. And this is, a huge ensemble type of models. So there's various methods with different parameterizations. So you can imagine, when you think about classical AI, I mean, I don't want to give away the farm, but it's vaguely the 5 or 6 most common methods that you would know. Those are the ones that we use as well. And then the aggregation is kind of fun as well, because apart from the ensemble itself, which you can do a majority vote sort of by bagging all the outcomes, you can have You can have also boosted results, which have to do with, since you've internally validated each and every one of your models, if one of them outperforms the other, then you can give it a slightly different weight. So there's different ways of scoring this. So that's one thing. Once we have this, we also have a threshold for what is enough for us to say that we believe that we have predicted a new property. So let's say you had 100 models and 51 agreed. Is this enough, or do you want 90 to agree, and so on? And then the second round, which has to do with how sort of how likely something is to be important, isn't only for the predicting the new property, it's whether or not this property is relevant to the particular disorder, which has to do with the hypergeometric that we apply for the enrichments. I hope I kind of answered it.
I think you answered most of it, but I would like to expand a bit on this. You mentioned ensembles and very large ensembles, but you also mentioned interpretability. How do you preserve explainability in very large ensembles?
That's a very good question. So I think I didn't use the word interpretability, but yes, you can indeed do it. It's just it requires a lot of reverse engineering, right? So what I mean by this is that you could have, for example, a model that uses 100 features, and you can have 1,000 of these models that say something. So if you want to, you can go and have a look at individually each of the 1,000 models, right? But that would be hugely time consuming. So what you can do instead is look at the saved models, look at the features that were used, look at the weights, look at residuals, look at stuff like that, and see, or even at the pre-processing stage, by looking at the information content, you can kind of see which were the more important variables that guided towards a particular prediction. So again, depending on what we're trying to do, we can give it, for instance, if you're talking about structural properties linking to a particular biological activity, then when a model tells you what type of structure you're looking for that particular activity, then you can go back and look at which features were important. So the same goes for any type of descriptor, by the way, but I just thought that this would be a sort of a more down-to-earth example.
So the next one is about the cost of clinical trials. How would it compare to the computational costs of in silico trials?
That's a very difficult question.
Have you thought of in silico trials to start?
Can we qualify in silico trials? Because I've got several different definitions for this, and it's a little bit of a gray area. I don't have the comparison, no. But if we qualify it a little bit, maybe in the chat or what have you, then it might help me sort of venture a guess.
One thing that we know about in silico trials is that at the moment you cannot use them to get an approval, right, for any therapy. So yeah, even if they're cheaper, then still they're not there yet.
I mean, it's sort of the same. Talking about AI, it's sort of the same thing between the internal validation and the prospective validation. I don't know if there will be a point where you'll be able to do a purely in silico trial. Maybe if there is so much overlap in every aspect between a prior trial and the current trial in terms of, say, some behavioral aspect or something, you might be able to, but otherwise, well, yeah, you're absolutely right. I mean, you don't have any approval to date. I don't know if and when you will have any. And to be perfectly honest, I mean, I wouldn't be able to comment on the cost as well.
Next question. Where is the bottleneck for a company working in drug repurposing? New algorithms to get higher accuracy, or is it finding a good clinical partner?
That's a good question. It's a little bit of everything. So it's a multi-parametric thing. I mean, every in silico company that you talk to will say that they have great models, and we do actually have internal validation statistics which will suggest, both in terms of predicting the new properties, when we hide away some data and we repredict them, we get a very good average accuracy, and the same goes for even predicting stuff directly to the clinic. So we work end to end. So as soon as we make a prediction, we can then go and see, once you've hidden the data, you can make sure you can validate it internally. So we've been able to sort of get a lot of the stuff that is already in clinical trials as a validation. Now you might say, well, maybe those will not work. Yeah, okay, but at least up to the point where something went into clinical trials, we do have that validation quite decently. So yes, definitely, you know, if you can get your accuracy from 80% to 90% or higher, that's fantastic. But bottlenecks include costs, so you do have to have the assessment. We do have it early on in our filtering sort of node, so that's one thing. Another thing that you have to have a look at is all of this feasibility. So can something does it have an existing animal model, for example? That's something very important to us, or at least some method which will definitively on a preclinical level say that you can indeed have an effect in this particular disorder, and then use this as an argument for the human intervention. So the established preclinical work is also very important. Good clinical partner is sort of very much dependent on the scope, so whether it's rare or a common disease, and then resources. So in the rare diseases, definitely you do want a good clinical partner. In more common disorders, because there's lots of good clinical partners, I'd say no, but then you require a lot more resources. So usually a company like us would have to collaborate or hand it off and pay somebody externally, a CRO, and they will have to conduct a multi-center trial and then aggregate all the results and what have you.
Thank you. The next question is about funding and sponsorship. I wonder if you could share more regarding the funding scheme approach. I mean, beyond R&D projects, who would pay, at which stage, who would initiate and fund the search for a new drug candidate?
Right. So again, multiple answers and very good question. We are funded by a VC and several grants, right, which have come from a variety of locations. Who would pay this, right? So in the rare disease sort of aspect of this, you could receive grants from patient networks, and you could, if you were granted orphan designation, have a particular access to a different pool of grants. You can also try something hybrid, so not necessarily academic funding, something like the EIC Accelerator, for example, which does fund companies in either as grants or in lieu of equity, or a hybrid model. And I think that probably the more reasonable one would be to go to a venture capital firm, which again can be something that is, if you're early stage, you could probably get away with going to a horizontal one that invests in multiple startups. The later you go, the more vertical you'd want somebody that's completely into health. You also have sort of these CVCs, or what, I think that's the term for them, you know, from pharma companies, which could also partner with you. I think the idea is getting your foot in the door. So if you get your foot in the door with somebody who's interested in the research, so a patient network, a pharma company that you're doing something with one of their drugs or some area of interest of theirs, or something like that, then once you get your proof of concept, then all of a sudden your network does tend to grow a little bit. But I think who would pay, I'd go with probably grants, VCs, and in sort of other very rare cases, maybe foundations, because, you know, you have people who have left a lot of money to charity. You know, maybe you go to the Bill and Melinda Gates Foundation, or maybe you go to the Michael J. Fox Foundation if you're doing Parkinson's, for example. So it's very much case dependent, but yeah, also very competitive, dare I say, all of these things.
Indeed. Thank you, George. And now we're turning to a more technical question. Concerning the data sources, you mentioned only open data repositories. Mm-hmm. Do the unstructured sources include literature, NLP analysis, natural language processing? And in terms of technology, if this is not, if you can reveal that, mm-hmm. Do you use knowledge graphs and related knowledge graph analytics?
Right. So start with the first one. Let me just scroll down to it. So yes, open data repositories, that's very true. we're talking about databases that are curated. We do apply further processing, right? So we don't just blindly bring in data. We look at databases that we trust, and we also look at factors for which would either let us keep or not particular data. So in terms of literature NLP, yes, but we have NLP from a particular source, which doesn't include all literature. If you want to go to something much, much larger, such as PubMed, then, for example, you'd have to, well, you'd have to basically say that I can't mine everything in every single PDF, so you'd go with the abstracts, right? So again, in both of these scenarios, you are missing some information, but at least it might point you into the right direction on where to look, you know, because you're trying to make the possible 200 papers that you're going to read into, condense them into about 10. In terms of knowledge graphs, we are currently talking with a particular partner, which again, because we're in the CDA phase, I can't really disclose, but we are talking about them bringing knowledge graphs into it. So yes, but not yet.
If I may, what is the use of knowledge graphs that you see? How do you intend to put them in your pipeline? Why are they useful?
So it will be for let's see, how can I say this without giving away something confidential? It will be about making sure that our predictions are indeed novel. So we want to make sure that we can find associations in both scientific literature and patents, which would, with a scoring system, be able to tell us whether or not something is novel or not. So in other words, if somebody's already touched upon something, we shouldn't go there because we wouldn't be able to protect our IP. We wouldn't be able to keep going with it.
Very different from the uses that we do of knowledge graphs. I think, you know, this is an interesting perspective. It's not so much of a cost comparison anymore between the in silico clinical trial and the actual clinical trial, but do you see in silico trials as an interesting filtering step in your pipeline?
You could use it. There is merit to use it as an extra de-risking factor. Sure.
Have you tried it?
Not currently, no.
It sounds interesting, especially if the cost is not very high, which I don't think the cost of in silico trials are particularly high. Getting them right is difficult, but running them is not necessarily a problem. I would like to thank you again very much, George, and I think the discussion was really very interesting and very coming from very different perspectives. Also from our side, as an AI lab, I see a lot of potential for collaboration, and we've talked about that before. I hope we'll get to that.
We can actually act on it. Yeah, absolutely. Yeah, and thank you very much for having me and the opportunity to present the company. It was very nice. And thank you all also for the questions. You know, the questions sometimes, you know, we're not able to answer them, especially if there's something confidential, but usually they are very interesting in probing. You know, we also get feedback, and we can this feedback can be used to integrate stuff into both the thought process and the pipeline itself. So again, thanks to all.
I'm really delighted by the fact that you started the clinical trial, and I hope that soon enough there will be a new treatment for Fragile X patients, and a lot of successes for you.
Thank you so much.